Hey everybody, so this video is going to cover if you are confused on this first summative target. So this first summative learning target is I can use and analyze Punnett squares to calculate the possible phenotypic and genotypic ratios of offspring in a monohybrid genetic cross. Basically, can you do a monohybrid Punnett square and can you interpret that data? Can you tell me what all of that means? In order to be able to do that, you need to know what an allele is. You need to know what homologous chromosomes are. You need to be able to identify genotype. So is it homozygous dominant, homozygous recessive, or heterozygous? And you need to be able to explain how genotypes are expressed as phenotypes. So again, this video, we're going to review this target, learning target sum 7a. So if we go back to our introduction to genetics, I'm going to skip all of the stuff about Mendel and just get into the nitty-gritty and the things that we need to know. So first off, what is a genotype? A genotype is a person's genetic code. So if you look up here, we have 2 little y's, 2 big y's, and 1 big y, 1 little y. This code, this genetic information, is the genotype. And it codes for the physical expression or the characteristics that we see. What we see is an individual's physical appearance. That's their phenotype. So the genotype, the code, codes for the physical expression. So here in this example, we've got 2 little y's, and that codes for a green pea plant. 2 capital Y's codes for a yellow pea plant. And a big Y and a little y also codes for a yellow pea plant. Now, an allele is a version of the same gene. So we've got 2 alleles here, a big Y and a little y. Both of them code for the color of the pea plant, but it's 2 different colors, 2 different alleles. Little y's code for green and big Y's code for yellow. Now, it's important to remember that there is a difference between dominant and recessive. The dominant allele is indicated by a capital letter, so that capital Y, and that's going to overpower or overshadow the recessive allele. The only time I'm going to see the recessive allele is if I have 2 of them. So the only way to get a little green pea plant in this picture is if this little pea plant inherits a little y from dad and a little y from mom. Heterozygous and homozygous, the difference is heterozygous is different. Hetero means different, so when you're talking about a heterozygous genotype, you're talking about 1 dominant allele and 1 recessive allele. Homozygous, homo means the same, so when we say homozygous, we either have 2 little alleles or 2 recessive alleles, or 2 dominant alleles. So long as they're the same, it's homozygous. You can be homozygous recessive or homozygous dominant, or the third option is heterozygous, and that's 1 of each. All right. Now, when we fill in Punnett squares, we start off by first needing to know what the parent's genotype is. Once we have the parent's genotype or their genetic code, we can take these 2 alleles from 1 parent, and remember the law of independent assortment and the law of segregation. So the law of segregation says that these 2 alleles are going to separate into the gametes. So let's say this is mom. Her 2 alleles, 1 big T is going to go into 1 egg cell, and the little t is going to go into another egg cell. If this is dad, this big T will go into 1 sperm and this little t will go into a different sperm. So they have to separate. They cannot stay together. So I'm going to take the first parent's alleles and put them on the top boxes. Remember, it does not matter which parent goes on top and which goes on the left, so long as the alleles that are together stay on the same side. Then I'm going to take the next parent and put them over here on the left. Now, in order to fill in this box, I'm going to take the allele that's on top on the outside and bring it down and the allele that's on the left and bring it over. So this box here would be big T, big T. Box number 2 would be big T, little t. Box number 3, big T, little t. Notice how these are written the same even though we would have put the little t first. When we're writing genotypes, the capital letter always comes first. It doesn't change what it means. So if you put the little t first, it means the exact same thing. It's just a formality to put the big T first. Box number 4 is going to be little t, little t. So once I'm able to fill this out, I've completed half of the first summative learning target, right? I can fill this out. However, if I don't understand what any of this means, then it's not going to help me. So what I need to be able to do is look at this and figure out the genotype ratios and the phenotype ratios. So if I say that T codes for height in pea plants, big T means tall and little t means short, tall is dominant, thus indicated by the capital letter. I need to be able to say what the probability of the next generation's height ratios would be. So here I have 2 big T's. This is homozygous dominant. So this individual is going to be tall because they have 2 of the dominant alleles. I have 2 of the same genotypes here, big T and little t. These are heterozygous genotypes. And because they have that dominant allele, these 2 phenotypes, so the physical expression of this genetic code is also going to be tall because they have that dominant allele. Box number 4 down here has 2 little t's. So the phenotype or the physical expression of this genetic code or this genotype is going to be short because I have 2 recessive alleles. So my genotypic ratio is 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive, or 1, 2, 1. That is my ratio. My phenotypic ratio, 3 out of the 4 are going to be tall because 3 out of these 4 boxes have a capital T. So 75% tall and 25% short, or a ratio of 3 to 1.